the last of our blood cells that we'll talk about are platelets, which I had started talking about in our last lesson. So blood platelets function in hemostasis, which is the cessation of bleeding. If we can't stop bleeding, we could potentially bleed out or hemorrhage, and this could, well, if you lose your blood, then you're going to lose all of your energy and your ability to do work, and you die. So it's super bad. So platelets are very important. There are three mechanisms to the hemostatic, or to hemostasis, that's what I want to say. Three basic functions. Vascular spasm, where the blood vessel itself squeezes so that you have less blood moving to the site of damage. A platelet plug formation, where all the platelets start to aggregate at the site of the damaged tissue, and the platelets plug up that spot. And then all of the proteins associated with coagulation come in and form a blood clot. So the first step is vascular spasm, where on each side of the damaged tissue, you're going to cause the blood vessel to constrict. Smooth muscle in the blood vessels will contract, causing constriction. And then we have platelet plug formation. So normally the walls of our blood vessels are covered with a special chemical called prostacyclin. And this chemical keeps platelets from adhering. So the platelets will kind of breeze up and down along the blood vessel line, and they won't attach. But if we have damage to the blood vessel, so something cuts the blood vessel or something internal occurs that causes damage to the wall of the blood vessel, platelets can attach. And so the platelets will, if they feel any like exposed collagen fibers, they'll attach to those collagen fibers, and immediately they'll start secreting chemicals. Those chemicals will cause more platelets to move to the site and release their chemicals. This is called a platelet plug. It is, hold on, a positive feedback mechanism. And so I found this figure, I just liked it because it shows that positive feedback loop. So something caused damage to the blood vessel wall. This activates platelets to move to the site of the damage. The platelets immediately start to release chemicals. These chemicals send information out to cause more platelets to move to the site of the damage, and more platelets will adhere to that same region, and then they release their chemicals until that entire area is closed up. At that point, there's no more, or there's no spot for the platelets to attach, and the platelets no longer release the chemicals. So this is a positive feedback loop. It's positive because we're pushing farther and farther instead of moving it back. As more platelets move in to that area, they send signals out for more platelets to move in. That's positive. Makes sense? And so here we have that platelet plug formation. And then we have the last step, the most important step, called coagulation. This is where proteins in the blood plasma get activated and form a clot, a protein fiber clot, where the platelet plug was. So this can be activated in more than one way. So there's an extrinsic pathway and an intrinsic pathway. The proteins that are produced, so the proteins produced for blood clotting are produced by the liver. So the liver is very important in this function. Without the liver, with liver damage, then you're going to be more likely to not be able to produce the proteins, and you're not going to be able to produce a blood clot properly. Make sense? So things that we need for blood clotting to occur, we need the clotting factors produced by the liver. We need the fibrin, which is also produced by the liver. We need certain chemicals, calcium, platelet, I already mentioned clotting factors, vitamin K. These are all important in helping to produce the clotting factors, as well as in helping us to form the blood clot itself. So there are two different mechanisms that can activate the coagulation cascade, the intrinsic pathway or the extrinsic pathway. Intrinsic pathway starts with damage internally to the blood vessel itself, whereas extrinsic is started by damage that came external to the blood vessel. So here would be extrinsic. You're cutting, well, in this case, carrots, and you cut your finger. That's external damage. You cut the blood vessel and you start to bleed. Intrinsic damage would be caused inside the blood vessel. So something in the blood vessel caused damage to the vessel wall. In both cases, it can cause us to bleed. And this leads to a cascade effect. So here you see factor 12, which is inactive. When it comes in contact with the platelets, becomes active, or activates, I should say, factor 11. Factor 11 activates factor 9. Factor 9, in the presence of calcium and platelet factor 3, activates factor 8. So do I care about this? No. And I don't care about this either. What I do care about is this common pathway and how it leads to coagulation. So in both cases, we're going to, whether you're going extrinsically or intrinsically, we're going to activate factor 10. And factor 10 activates prothrombin activator or prothrombinase. Prothrombin activator activates, or I won't say activates, converts prothrombin into thrombin. Thrombin converts fibrinogen into fibrin, and fibrin is what produces the fibers of our blood clot. That's what I really care about. I do have a video I like to show. Again, don't worry about all the different factors. You just really want to know this part down here, okay? Come on. Always allow.
Damage to small blood vessels and capillaries frequently occurs. When these vessels are damaged, there are three basic mechanisms that promote hemostasis, or the stoppage of bleeding. Following damage, there is an immediate reflex that promotes vasoconstriction, thus diminishing blood loss.
I'm playing with my disease ball. It's called an infectious disease stress ball. Oh, but there we go. I love it. Sorry.
Exposed collagen from the damaged site will promote the platelets to adhere. When platelets adhere to the damaged vessel, they undergo degranulation and release cytoplasmic granules, which contain serotonin, a vasoconstrictor, and ADP and thromboxane A2. The ADP attracts more platelets to the area, and the thromboxane A2 promotes platelet aggregation, degranulation, and vasoconstriction. Thus, ADP and thromboxane A2 promote more platelet adhesion, and therefore more ADP and thromboxane. The positive feedback promotes the formation of a platelet plug. The final hemostatic mechanism is coagulation. Damaged tissue releases factor 3, which, with the aid of calcium ions, will activate factor 7, thus initiating the extrinsic mechanism. Factor 12 from active platelets will activate factor 11, thus initiating the intrinsic mechanism. Both active factor 7 and active factor 11 will promote cascade reactions, eventually activating factor 10. Active factor 10, along with factor 3, factor 5, calcium ions, and platelet thromboplastic factor, PF3, will activate prothrombin activator. Prothrombin activator converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin initially forms a loose mesh, but then factor 13 causes the formation of covalent crosslinks, which convert fibrin to a dense aggregation of fibers. Platelets and red blood cells become caught in this mesh of fiber, thus the formation of a blood clot.
All right. The last thing we'll talk about then are some disorders associated with blood clotting. One of these disorders is known as hemophilia, and this is probably the most widely known blood clotting disorder. Hemophilia is the inability to clot blood, and it's typically associated with the inability to produce certain protein blood factors. So if you don't have all of those clotting factors, then you can't produce the blood clot because the cascade is not going to work. Does that make sense? And if it doesn't, ask me because, well, obviously I don't know because you're at home or wherever you are watching this and I can't see if it makes sense. So hemophilia is an X-linked recessive disorder. What that means is females tend to be carriers, but they tend to have one normal allele as well, so they won't show the disorder. Females are normally either heterozygous or they're homozygous dominant where they don't have the disorder. Males, on the other hand, if they are born from a mom that is heterozygous, they have a 50% chance of having the disorder. Another disorder is called thrombocytopenia. So in this case, you're still not going to be able to clot blood properly, but the reason is because you don't produce platelets properly. This can be associated with different infections as well as with cancers. HyperCoagulation is the opposite, where you're clotting blood in blood vessels that you shouldn't be clotting blood in. This can lead to an embolus. So an embolus is something where a blood clot that has formed in the blood or unattaches from the wall and starts to move through the blood vessels, and it can clog up different arteries, causing us to have problems with breathing, inability to get blood to important organs like our brain, and it can eventually lead to death. So here is what an embolism in the lungs might look like. So you have this large blood clot in the blood vessels, and blood can't move very effectively. And that's going to cause breathing issues as well as eventually it could cause death. Here's an embolism in the carotid artery, the internal carotid artery, I think that's showing because it's going up here. And if it's blocking blood going either to the external or internal carotid artery, it's blocking blood from going to the brain, and that can lead to a stroke and eventually death as well. All right, that is the end of this lecture. So I'm going to stop this, and I hope you have got a lot of information that you can come to class and ask me questions on. Bye.